Hello everyone, welcome to the next lesson in the animal behavior course. Today we're going to be discussing different hormonal mechanisms and how they influence behavior. So hormones are molecular messengers. They affect many traits, including behavior, both directly or indirectly through changes in cell metabolism or DNA expression. Hormones work at really low speeds of response, several minutes to several hours for these changes to take place and for the behavior to occur. Now, for the most part, they're produced and then secreted into the bloodstream by these ductless glands. Now, there are a few exceptions to that case, but we will sort of ignore those exceptions for now. Many glands of the endocrine system also secrete more than one type of hormone, and the same hormones can have different effects on different target cells, so that the correct receptor site for the hormone is required in order for the hormone to have an effect. And so basically what this is saying is that all hormones won't work on all cells and all hormones don't have universal effects. It depends on where they're binding in order for them to sort of have that effect. So how do we test if hormones are causing behavior or they're resulting in behavior? Well, let's talk about one of the very first experiments in behavioral endocrinology. This was by Berthold, and it's known as Berthold's chicken experiment from the 1800s. And so farmers had known for centuries that castrated male animals behave differently. They're less aggressive, they don't develop the same secondary sex characteristics, and they don't show normal sexual behavior. Berthold tested this directly with chickens to understand what role testosterone was playing. So he removed the testes from young male chickens, and then he found that those birds grew up without the big comb and sort of the wattles and the crowning, and they also didn't display normal courtship or aggression behavior. And so, in short, no testes meant no typical rooster behavior. Now, here's the clever part, though. Berthold took a testy and then he put it back inside the body cavity of castrated males. And even though it wasn't connected to nerves or anything else, those birds still developed into normal roosters. And so what was the conclusion? Essentially, Berthold found out that the testes weren't actually sending signals through nerves or to the brain. What they were doing was actually releasing a substance in the blood. We know that substance today is testosterone, but at the time you didn't actually know that. And so we call these substances, these messengers, hormones. And so this was the first clear demonstration that hormones can affect and control behavior. So hormones are commonly manipulated nowadays in a variety of species to test a variety of different things. And they're all used to determine whether the hormone is sort of the root cause of that behavior. It is in some instances, it's an easy thing to manipulate. So for example, you can remove glands upon birth, and so that will eliminate the hormone source. This is through things like maybe like a castration or an adrenalectomy or like an ovarectomy, right? So you can remove the gland entirely and then see how the organism develops and how it behaves later in life. You can also reimplant the glands and try to restore normal hormone function. This doesn't always work, but through surgery and through complex medical practices, which we don't have to know for this course, you can get glands back into an organism so that it can continue producing those hormones. You can also do non-surgical manipulation. So you can block receptors through antagonists, through drugs that bind to the receptors, but then block them from letting the hormone bind. You can also add in hormones. So just like you can take a hormone pill as a human, you can take pills as an animal, and you can feed hormones to animals to up their hormones of certain levels and then see how the introduction of certain hormones can change behavior. And so this brings up a really important distinction. As we're going to be doing these experiments and going over them, you're going to get some hormones that are required, some hormones that just affect behavior. Hormones can kind of do a range of different things. And so it's really important to make this distinction between the word necessary and the word sufficient. And so they're really easy to mix up, but they do mean very different things. So if something is necessary, it means that the behavior won't happen without it. For example, if you remove testosterone in roosters, they won't crow, they won't fight. And so this shows that testosterone is necessary for those behaviors. If you can remove it and the behavior stops, then that hormone is necessary for that behavior to occur. Now, sufficiency is a little bit more tricky, a little bit harder to show. So if something is sufficient, it means that just by receiving that hormone, that alone is enough to make the behavior happen. So for example, testosterone implanted in castrated roosters brings back crowing and aggression, and showing that it is sufficient to induce these traits and behavior as well. But we'll come to find that that's usually not the case, that there are usually instances where getting something to prove to be sufficient is very, very difficult, because there are usually so many more complex factors at play than just whether the hormone is present or not. And so it's important to know that hormones are not always both. Sometimes a hormone is necessary but not sufficient, and sometimes it's sufficient but not necessary. And that's why experiments often combine both the removal, replacement, and supplementation to sort of tease these different explanations apart. And so in the next couple examples, we're going to go over when hormones are necessary but not sufficient, as well as when hormones can be sufficient but not necessarily necessary. So let's go over this first example. In the male anolis lizard, one of the most striking behaviors is this dewlap display, which you can kind of see right here. That bright flap under the skin or throat will kind of puff out and go back in. And males use this display during mating season to both attract females, but then also to defend their territory against rivals. And so what's important here is that with the dewlap display, it only appears when the testes are active in the spring. If the testes are inactive or removed, then the display disappears. So is testosterone causing this behavior? Is it necessary or is it sufficient? Well, let's run an experiment to find out. So what we do is we ask the question, is testosterone necessary and is it sufficient to have the dewlap display? And so what we did is we took males and we castrated them at a certain point, and we measured over time the number of dewlap displays that they displayed. And so before castration, you're getting a lot right here. You can see down by the really high start of the graph, they're having a lot of dewlap displays. And then once castration happens, it falls off to nearly zero, right? And then that stays at zero for quite some time. And then all of a sudden, if you re-give testosterone back to the anolis lizard, it will then begin doing the dewlap display once again at a high frequency. And so my question is this demonstrating that testosterone is necessary? And does it demonstrate that it is sufficient to cause the dewlap display? And so if you need a second to think about it, go ahead and pause the video. But the answer is actually a bit of a tricky one. We do not know. There is nothing to control or to compare it against. It could be, right? So this graph looks really great, suggesting that maybe it is necessary and maybe it is sufficient. But the problem is we did not run a control condition, right? So it's possible that through castration, which is a surgery, maybe the surgery is so traumatizing that it prevents them from then doing the dewlap display again. Or maybe addition of testosterone could be like a placebo effect, causing them to do the dewlap display more frequently, but without a reason for there being testosterone. So it's kind of a trick answer, and I wanted to show this to you guys because it's not as straightforward as this graph would make it seem. We need to be able to have a control condition in which we do a sham operation and give a placebo pill to make sure that this is what's actually happening. And so by adding a control group, we can do a sham operation. And a sham operation, all a sham operation is you pretend to put the animal through surgery, you mess with it, you put it under anesthesia just like you would for castration, but you don't actually remove the testes. And so basically then that eliminates the possibility that a deduction in dewlap displays is the result of surgery. And instead, we know through that control by this difference here, right? So the sham operation anolis lizards continue to do the dewlap display, which means it is indeed the removal of the testes and the lack of testosterone that is causing the reduction in the dewlap display. It's not just the fact that they're going through surgery. And similarly, we see that the testosterone pills will restore dewlap display, but placebo doesn't have any sort of effect on the dewlap display. So once again, that's showing that it's the testosterone that matters. And so in this case, testosterone would be both necessary and sufficient. Necessary because if you remove it, the behavior stops, and sufficient because just reintroduction of testosterone causes the Anolis lizard to engage in the dewlap display even more. And so let's move on to a different example. So here we're going to discuss sexual behavior of rats. And so rats engage in two different types of behaviors in order to reproduce. First, the male will mount a female, but reproduction is not possible unless the females also engage in this sort of arching behavior where they arch their back, and this behavior is known as lordosis. And so normally males will never perform lordosis, and normally females will never ever mount. So this automatically should be ringing bells in your head to go, okay, if males are doing one thing and females are doing another, this is probably a hormonal mechanism, right? Because males and females have different hormones acting in their bodies, both during sexual maturity, but then also into adulthood. So what might it be? Well, there was actually a series of experiments that tested this very thing to see what was causing males to mount and what was causing females to engage in this lordosis behavior. So this is the normal trajectory of mice. You start out with a pup up here at the top of the screen, and normally males will mount in adulthood, and normally females will do lordosis into adulthood. Right, I do want to point out these baby rats at the top, they're not dead. I promise you this is what a baby rat looks like. Rat pups are just very altricial and they need a lot of help from their mothers, just kind of like baby birds, right? Honestly, kind of like humans too, right? Humans cannot really function on their own without their mothers. So similarly, rats are the same way. So let's sort of look at the trajectory and the experiments that they did with males first. So here are the various experimental manipulations that they did, as well as the outcome. So the first two are pretty straightforward. So the first one, if you castrate a rat later in its life, after it's reached sexual maturity, it will still mount, right? And secondly, if you do an early castration on a male pup, but then give it a testosterone implant, it will also once again mount. Now, the key manipulation here is if you castrate a male pup early and do not supplement it with testosterone, then a male rat, when it reaches adulthood, will engage in lordosis behavior. And so this is showing that during a critical period, testosterone can induce male mounting behavior later in life. And so testosterone, when it's present early in the critical period, then when the rat gets to adulthood, it will then mount. But if you remove that testosterone during that critical period, the male rat will then behave more like a female rat and engage in lordosis rather than mounting behavior. All right, so now let's look at the females. So the first thing that they did was they took females and they transplanted testosterone into a female pup. And what do you know? Lo and behold, the female pups engaged in mounting behavior once they reached sexual maturity. But what happened? What are the other manipulations that they ran? Specifically looking at the hormone estrogen. So now we have sort of complex results. If you remove the ovaries during a critical period, then you remove estrogen from their system, and they actually won't perform any sort of sexual behavior whatsoever. They won't mount, and they won't engage in lordosis. They won't mount because they don't have testosterone, and they won't engage in lordosis because they don't have estrogen. And so that's what removing estrogen through an ovarectomy does. And so, well, if they were behaving like males, then what would we suspect? We would suspect that if we remove the ovaries and then give them estrogen, that then they would do lordosis, right? But that's actually not the case. That is not what happens with female rats. If you overrectomize them and give them estrogen, they still will not engage in lordosis behavior. They will still do no sexual behavior whatsoever. And so here, real quick, this is showing that estrogen is not sufficient to induce lordosis, right? There's something else that has to happen for lordosis to occur. You cannot just remove estrogen and replace it, right? You can replace estrogen, but something else has to happen. So if you actually take a mouse or a rat that has had an ovarectomy and you've given it estrogen, then you also give it progesterone, and as well as touch its back, then the rat will engage in lordosis. And so it's not just estrogen, but it's a combination of other factors that induce lordosis. And so my question to you would be, is estrogen necessary, and is it sufficient to induce lordosis in female rats? The answer is it's necessary, because if you remove it, it does not happen. But it's not sufficient, because there are other factors that are needed in addition to estrogen in order to induce a lordosis behavior. And so I know this is a bit of a complex result, so let's go over the main key takeaways. First, testosterone during a critical period is necessary to produce mounting behavior. We know this because if you remove testosterone, male rats will not mount. Second, testosterone during a critical period is also sufficient to produce mounting behavior. And why do we know this? By simply adding testosterone to females, they will then mount later in life. Third, estrogen is necessary to produce lordosis, because if you remove estrogen, you no longer get lordosis. Fourth, estrogen is not sufficient to produce lordosis, because if you add estrogen, it does not cause lordosis. There's a bit of a typo there on the screen. If you add estrogen, it does not produce a lordosis behavior. So I'll make sure I fix that before I publish the video. And also, basically, we know this because there are other hormones and also physical touch is needed. So sufficiency is not met for estrogen to cause lordosis. All right, let's go over one more example in rodents. This is talking about the hormone oxytocin. Oxytocin is a very complex hormone, often involved in maternal care, but also in affiliation in general. And so here are two manipulations that they found. First, if you inject oxytocin into the brain of a virgin female rat, one that has never had any offspring, that injection will trigger maternal care in the virgin rats. So rats that have never had children will all of a sudden be induced to nursing them and taking care of pups, licking them, etc., etc., just simply by adding oxytocin into the system. However, if you take oxytocin away from experienced rats that have had offspring already by blocking the receptors, those female rats, those mom rats, will still engage in maternal care behavior. And so what does this say about oxytocin? Is it necessary, and is it sufficient? Take a second and think about that. Pause the video if you need some more time to think about it. This is actually an example of where oxytocin is sufficient, but it is not necessary. For example, we know this because by giving a virgin non-mother rat oxytocin, we can induce maternal care. That makes it sufficient. However, just by simply blocking oxytocin does not stop maternal care behavior from occurring in experienced moms. And this shows that it is not necessary. Moms with maternal experience will still engage in this behavior whether or not they have estrogen, right? Or oxytocin, sorry, not estrogen, oxytocin. So that means it's sufficient but not necessary. And so this is a very rare instance where that is the case, but in this case, sufficiency was proven but not necessity. Awesome. So that concludes the lesson on hormonal mechanisms. Hormones are a powerful way for behavior to be modulated because they act throughout the body and can coordinate changes across multiple systems at once. And what we've seen today is that hormones can directly affect a behavior. Sometimes they set a stage by making certain behaviors possible. Maybe they make them more likely or maybe more intense. But we also learned that hormones can be necessary without being sufficient, and in rare cases, they can be sufficient without being necessary, sort of depending on the context. Ultimately, the big takeaway is that hormones interact with the nervous system, one's environment, and the animal's internal state to shape their behavior. In the next part of the course, we're going to shift away from hormones to learn how animals learn behaviors and to see how these different mechanisms connect with Tinbergen's framework of explaining animal behavior. I really enjoyed this lesson, and I hope that this was very, very helpful.